अस्सलाम वालेकुम माय डियर स्टूडेंट्स। आज हम बात करने जा रहे हैं सेक्सुअल रिप्रोडक्शन इन बैक्टीरिया। पिछले वाले क्लास में हमने क्या देखा, हमने एसेक्सुअल रिप्रोडक्शन डिस्कस किया और वी टॉक्ड अबाउट डिफरेंट स्ट्रेटजीज ऑफ एसेक्सुअल रिप्रोडक्शन। टुडे वी विल हैव अ brief introduction about sexual reproduction, and then we will talk about sexual reproduction in bacteria and its different types. Sexual reproduction क्या होता है? It is actually an advanced mode of reproduction in living organisms where meiosis takes place, or you can say where formation of gametes occurs and where fertilization takes place or fusion of gametes occurs. Okay, so the prerequisite for sexual reproduction to happen is there should occur meiosis and there should occur fertilization of gametes. Another important point about sexual reproduction is in sexual reproduction, two parents are involved, and these two parents may be genetically different. Okay, while as in case of asexual reproduction, only one parent is involved in the creation of, or you can say generation of, new individuals. Okay, this is another difference between sexual reproduction and asexual reproduction. Now, what is the outcome of sexual reproduction? The main outcome of sexual reproduction is it leads to the recombination of parental genes. Hence, you can say there is generation of new varieties. How recombination of parental genes occurs? It occurs as a consequence of crossing over that occurs during meiosis and by the fusion of gametes. Fusion of gametes and crossing over that occurs during meiosis, it leads to the recombination of genes which forms new varieties, and because of the formation of new varieties, it will ultimately lead to the generation of new species, and you can say the evolution is occurring. This is the main significance of sexual reproduction. While asexual reproduction does not lead to the creation of new varieties, and hence we can say it does not lead to evolution, sexual reproduction leads to the creation of new varieties and species, as a consequence of which we can clearly say it leads to the evolution of new species and varieties. In bacteria, reproduction is not purely sexual, rather it is parasexual means of genetic recombination. अगर हम बात करेंगे sexual reproduction in bacteria, तो जो बैक्टीरिया होते हैं उनमें ना तो मियासिस हो जाती है, ना ही गेमेट फॉर्मेशन हो जाती है, यू कैन से, और ना ही fusion of gametes होता है. Okay, instead there is simple transfer of genetic material from one cell to another cell, but genetic material transfer is happening, and as a consequence of which you can say it is similar to sexual reproduction. That is why we say it is parasexual, besides sexual, or an alternate mode of sexual reproduction. It is not that much advanced mode of sexual reproduction which occurs in case of higher plants and higher organisms. Okay, and here, in sexual reproduction in bacteria, there is horizontal and non-reciprocal gene transfer. Say for example, these are two cells. This cell contributes genetic material to this cell, or you can say genetic material transfers from this cell to that cell. Okay, this is known as horizontal gene transfer, and this horizontal gene transfer, it is non-reciprocal. That means this cell at the same time cannot contribute genetic material to that cell. यहाँ से यहाँ की ओर जेनेटिक मटेरियल ट्रांसफर नहीं हो जाएगा, बल्कि यहाँ से यहाँ की ओर जेनेटिक मटेरियल ट्रांसफर हो जाएगा. दिस इज नॉट रेसिप्रोकल. ओके, now we know that two cells are involved, and these two cells, bacterial cells, they are mature individual cells. Okay, they are independent cells, and one is known as, एक सेल को हम बोलते हैं डोनर, दूसरे सेल को हम बोलते हैं रेसिपिएंट. ये जो डोनर सेल होती है, it donates a portion of its DNA. It donates a portion of its DNA, and that portion of DNA transfers into the cytoplasm of this cell, which is known as recipient. Okay, this portion of DNA which this cell donates, this is known as exogenode, and this exogenode, it gets assembled into the genome of this recipient cell, and the genome of this recipient cell, this is known as endogenode. याद रखना, two cells which are mature, they are independent cells. One is called as donor, and this donor cell, it donates a portion of its genetic material. Okay, this is known as exogenode, and this cell, which is known as recipient cell, it receives this exogenode and assembles this exogenode into its own genome, which is known as endogenode. Now, the transfer of genetic material in bacteria, it can happen due to a number of agencies, or it can occur through different routes. Okay, the first one is conjugation, second one is transformation, and the third one is transduction. These are different modes of sexual reproduction in bacteria. Okay, now what is the fate of exogenode? जो exogenode होता है, जो genetic material होता है, small fraction of genetic material which is donated by the donor and received by the recipient, what is its fate? इसके साथ क्या हो जाता है? What is the ultimate fate of exogenode? It can go through a number of processes, or you can say it can have a number of destinies. Okay, the first one is it gets integrated into recipient genome. यहाँ से genetic material transfer हो जाएगा इस वाली सेल में. Okay, and here it will get included in the genetic material of the recipient genome based on homology. ठीक है, याद रखना. The first one is this exogenode, it can get integrated into the recipient genome. ये recipient genome है, इसके साथ मिल जाएगा. Or it independently exists inside the recipient cell. या तो ये काम हो जाएगा, इसके साथ integrate नहीं हो जाएगा. It independently exists in the cytoplasm of the recipient cell. Okay, and here that cell having another portion of DNA from another cell which is not integrated into the genome, this is known as partial diploid. याद रखना, it can either integrate into the genome of the recipient cell or it can independently exist inside the recipient cell. Okay, or in some cases the exogenode, it is degraded in the host cell which is known as host cell restriction. यहाँ पे आप देख लीजिए, ये donor cell है, ये recipient cell है. Okay, it has its own DNA, और यहाँ पे एक फ्रेगमेंट यहाँ पे DNA का ये ट्रांसफ़र हो गया इस वाली सेल में. यहाँ पे हम दिखाएंगे. ओके, एंड दिस सेल, it doesn't either exist on its own in the cytoplasm, nor does it integrate into the host genome. Instead, this is degraded here because of the specific metabolism of the recipient cell. The recipient cell does not allow this foreign DNA material, or you can say the exogenode, to exist inside it. Okay, now we will talk about conjugation. First of all, conjugation was discovered by Liederberg and Totem in the year 1946 in Escherichia coli. Okay, in conjugation, there is transfer of genetic material through physical contact. When we say conjugation, it means the two cells, they must be in close contact with each other. Okay, they come in close contact with each other, physical contact. Okay, and this is usually mediated by a spatial plasmid called F factor or fertility factor. F factor or fertility factor plasmids, they contain about 20 genes, and most of these genes, they are implicated in conjugation. That means the plasmids F factors, they contain almost 20 genes, and most of these genes are involved in conjugation. वो conjugation में उनका role है. Okay, the F plasmids, they function to form a conjugation bridge or sex plus between donor cell and recipient cell. देख लीजिए क्या होता है. आपके पास दो सेल्स होती है, वन इज डोनर, अनदर वन इज रेसिपिएंट. इस वाली सेल में एक तो अपना जेनेटिक मटेरियल होता है, नॉर्मल जेनेटिक मटेरियल, इसमें भी होता है. ठीक है, लेकिन ये जो डोनर होता है, इसमें एक एक्स्ट्रा सर्कुलर डीएनए मटेरियल होता है जिसको हम बोलते हैं प्लाज्मिड. ओके, इस प्लाज्मिड का नाम है स्पेसिफिकली, इसको हम बोलते हैं फर्टिलिटी फैक्टर और एफ फैक्टर. ओके, और इस वाली सेल में जो रेसिपिएंट होता है, इसमें ऐसा कोई किस्म का प्लाज्मिड नहीं होता है, इसमें फर्टिलिटी फैक्टर नहीं होता है या सेक्स फैक्टर नहीं होता है. ओके, सो यू कैन से दिस सेल एक्ट्स एज डोनर बिकॉज ऑफ द प्रेजेंस ऑफ एफ फैक्टर inside it, and this cell acts as a recipient because it lacks the F factor. Okay, and F factor grants this donor cell the ability to form the sex pilus or conjugation tube. ये वाली cell फिर conjugation tube बनाती है because of the presence of this F factor. Okay, so in conjugation you have two different mating types that we were talking about. Okay, you have two different mating types. One is known as donor. It contains F factor and it is also called F positive cell. Another one is recipient. It doesn't contain F factor and it is known as F negative cell. याद रखना. Okay, now the process of gene transfer that occurs during conjugation. It occurs in a number of steps. The first one is the donor and recipient cells, they come in contact either directly or through sex pilus. पहले क्या हो जाता है, जो दो सेल्स होती है डोनर और रेसिपिएंट सेल, ये तो एक दूसरे के साथ साथ आ जाते हैं. ठीक है, ये एक दूसरे के साथ साथ आना it happens either due to the formation of sex pilus or they can directly come into contact. Okay, without sex pilus. Okay, याद रखना, they have to come in close contact with each other. Then a channel is established between two mating types. Okay, एक channel जैसा structure बन जाता है, इसको हम बोलते हैं sex pilus. The F factor in the donor, it undergoes breakage at specific site and the strand separation, and one of the strands passes into the recipient through the sex pilus. Okay, ये क्लियर हो जाएगा नेक्स्ट वाले स्लाइड पे. ओके, याद रखना, जो ये डोनर सेल होती है इसका जो एफ फैक्टर होता है ये ब्रेक डाउन हो जाता है. ठीक है, एक पॉइंट पे ब्रेक डाउन हो जाता है, फिर इसकी रेप्लिकेशन हो जाती है, एंड जब व्हेन रेप्लिकेशन टेक्स प्लेस वन ऑफ द स्ट्रैंड्स इट गेट्स ट्रांसफर थ्रू दिस चैनल इनटू दिस सेल. ओके, इनसाइड द रेसिपिएंट कंप्लीमेंट्री स्ट्रैंड इज सिंथेसाइज्ड. यहाँ पे एक स्ट्रैंड यहाँ से यहाँ की ओर चला गया, अब यहाँ पे एक ही स्ट्रैंड है. ठीक है, इट कैनॉट एग्जिस्ट एज अ सिंगल स्ट्रैंड. यहाँ पे इसकी रेप्लिकेशन हो जाती है, ये डबल स्ट्रैंडेड प्लाज्मिड बनता है. ओके, इनसाइड द रेसिपिएंट कंप्लीमेंट्री स्ट्रैंड इज सिंथेसाइज्ड एंड डबल हेलिक्स इज फॉर्मड अगेन. यहाँ पे ये डबल हेलिकल प्लाज्मिड बनता है. ओके, द रेसिपिएंट सेल नाउ गेट्स एन एफ फैक्टर एंड बिकम्स F पॉजिटिव. यहाँ पे F पॉजिटिव था बिकॉज ऑफ द प्रेजेंस ऑफ F फैक्टर. ये वाली सेल F नेगेटिव थी बिकॉज ऑफ द एब्सेंस ऑफ F फैक्टर. नाउ दिस सेल as well as this cell, they both contain F factors and hence we can say this recipient cell also becomes F positive because it got the F factor from this cell. See here, this is the diagrammatic representation of the process of conjugation that happens in bacteria. Here this is the main DNA, or you can say nucleide, and this is the mobile plasmid which exists in the cytoplasm independently of this whole normal DNA. Okay, this is you can say this is donor cell and this is recipient cell. Okay, this recipient cell does not contain the F factor, and because of the absence of F factor you can say it is F negative and this is F positive. Okay, now this donor, it forms a conjugation tube or sex pilus because it contains F factor. F factor grants it the ability to form the sex pilus or conjugation tube. ये वाला cell conjugation tube नहीं बनाएगा because it lacks the F factor. फिर क्या हो जाता है, कंटिन्यूटी इज मेंटेन्ड बिटवीन दीज टू सेल्स. ओके, ये जो इसका प्लाज्मिड होता है इट ब्रेक्स एट वन पॉइंट, यहाँ पे ब्रेक डाउन हो जाता है इसका, और जब एक स्ट्रैंड ब्रेक डाउन हो जाता है तो इसमें क्या हो जाता है, दिस स्ट्रैंड इट स्टार्ट्स मूविंग, द ब्रोकन स्ट्रैंड इट स्टार्ट्स मूविंग थ्रू दिस चैनल इनटू द रेसिपिएंट सेल. रेसिपिएंट सेल के अंदर जाता है. ओके, नाउ both the cells contain one strand of the plasmid. ये वाली सेल भी, इसमें भी एक स्ट्रैंड होता है, ये वाली सेल भी, इसमें भी एक स्ट्रैंड होता है. ठीक है, यहाँ पे भी रेप्लिकेशन हो जाती है. ड्यूरिंग द ट्रांसफर ऑफ one strand of plasmid into this cell, यहाँ पे replication continuously होती है. So at the end you will see this cell contains double stranded plasmid, and once the single stranded plasmid it reaches the recipient cell, okay, यहाँ पे भी फिर इसकी replication हो जाती है. It forms a double stranded circular plasmid, or you can say it contains a full F factor. Okay, so at the end you will see both the cells, they contain F positive factors. Okay, and now you can say that this act of conjugation, it has created an F positive cell out of F negative cell due to the transfer of plasmid, okay, from donor cell to recipient cell. Okay, याद रखना, at the end you can have two donor cells. Okay, ये भी donor बन जाती है because it has got the F factor from this cell. So this is all about the process of conjugation that occurs in bacteria. This is all for today. See you soon with the other strategies of sexual reproduction in bacteria. Till then, thank you and God bless you all.